Hey friends, this one was heck long, so I'm going to break it into two. So, this is quiz 4.0, and there's lots of stuff in this one. Well, the second part has most of the stuff that you have not seen before, so we'll talk about that then. The first half's not so bad. Let's do that one today. Ready? Okay. So, question one, why must technicians practice aseptic technique when culturing cells? I call it sterile technique in class. It just means being careful not to get contamination done. So, that's to ensure that you're growing pure cultures of your desired cells and not some cells you don't want to grow, like bacteria from your fingers or something. Which of the following techniques destroys all microorganisms? That would be the autoclave, which is called sterilization. So this is actually a hierarchy of sort of cleaning, where cleansing is at the bottom. That's like wiping something down. And then it goes cleansing, sanitation, disinfection, and sterilization. Sterilization means there's no living cells nor spores, viable spores left. It's game over for everything. When using a microscope on the highest power objective, which focus knob should you use? The focus knobs, which one should you use? You should use the one that moves it the least, which is the fine knob. A solution with a pH of 3.0 is how many times more acidic than a solution with a pH of 6.0? So remember, pH, it's kind of inverse, where the smaller the number is, the more acidic it is. And 3 versus 6, pH works on the log scale, so it's log base 10 scale. So 3, here, let's do it on the screen here. 3 to 4 would be 10 times, through to 5 would be 100 times, to 6 would be 1,000 times. So a solution with a pH of 3 is 1,000 times, 3 orders of magnitude, 1,000 times more acidic. Salt, such as sodium chloride or sodium acetate, is commonly used in procedures to extract plasmid DNA. Which of the following best describes the role of the salt? So we didn't actually we used a different protocol for doing our mini preps. A lot of these questions are using kind of like old techniques that have been replaced by more effective techniques, but we're just going to have to deal with it because it's just a test. And step one is to not worry about it. Okay, the positively charged sodium ions dissolve cell organelles. That's not going to happen. The positively charged sodium ions provide a cushion, allowing for faster centrifugation. Yeah, that's pure BS. The positively charged sodium ions neutralize the negative charge on the DNA. Yeah, you remember the sugar phosphate backbone has those phosphates with by the time they're linked up with other atoms, have a -2 charge. So DNA is negatively charged. That's why electrophoresis works. And that's why you want to add salt to that sucker. The next one says positively charged sodium ions degrade the bacterial cell wall, which they don't. You need like a detergent to do that. Why is plasmid DNA purified using anion exchange column chromatography? So column chromatography, that's where you use a column with a resin in it, and whatever it is that you're trying to purify from a mixture sticks to the resin, and then you use an elution solution to unstick it when you want to capture it all by itself. So why is plasmid DNA purified? Why is it purified? Okay, DNA is positively charged. I'm going to stop reading because DNA is not positively charged. DNA is negatively charged, and it binds to the positively charged resin, and it's eluted with increasing salt. Seems reasonable. DNA has a natural affinity for most columns. Yeah, no. Plasmid DNA is small enough to bind to the column. So this is actually true in a different kind of chromatography called size exclusion chromatography, but we're talking about the key word here is ion. So we're going to look for something that has to do with charge. DNA is negatively charged. That's the best answer. A technician is testing the purity of a DNA sample using a spectrophotometer. He gets a 260 nanometer reading of 0.51 and a 280 nanometer reading of 0.65. Recalling that the preferred range for the value is 1.8 to 2.0 is the purity of the sample acceptable. Okay, well, here's the deal. We haven't done this. We have the qubit fluorometer, which we use to measure the concentration of DNA in a solution, and that's new, and that hasn't been around for very long. So they used to just use a regular old spectrophotometer. And if we go like this, and we say that this is absorbance over here, which is how much light gets absorbed, because remember, the way a spectrophotometer works is you have your sample like this, and you have a light source, a lamp, that shoots light through it. And then you have an electronic detector that I can like represent with an eyeball right here. And it detects how much that light gets through. And that happens at all sorts of different wavelengths. So that's what goes down here is wavelength. So two important wavelengths for testing the purity of DNA are 260 nanometers and 280 nanometers. And let's use this orange marker right here to say that we were going to put some protein in there. If we put some protein in there, we'd see a big spike of absorbance at 280 nanometers. Let's use this green one for DNA right here. If we put DNA in there, we'd see a big spike at 260 nanometers. So a pure sample of DNA would absorb a bunch of light at 260 nanometers, and a pure sample of protein would absorb it at 280 nanometers. So let's say we did it, let's say you made some human insulin using like bacterial cells or something, or yeast, and you wanted to purify that. No, you don't want to purify insulin. Sorry. You want to purify some DNA from it. So you want to take some cells and purify some DNA. And let's say you're going to use it for something downstream and you need it to be super pure. You don't want a bunch of other cell products like proteins in there. So what you do is you put that sample in there. This is absorbance, this is wavelength in nanometers. You put that sample in there, and what you do is you take this number, which is the absorbance at 260, and I don't remember what they said it was on there. We'll check it out. And then you take this number, which is the absorbance. So you take this number, which is the absorbance at 260, and you take this number, which is the absorbance of 280, and you find their ratio. So you take the ratio of the absorbance at 260 divided by the ratio of the absorbance at 280, and that's going to give you a number, some relative number that's going to tell you the purity of your sample. So the higher this number is and the lower this number is, the bigger this number is, and the more pure your sample is going to be. You want this number to be relatively big. And there in the test here, they tell us how big they want it to be. So they say that they want this number, I don't know, so they want it to be between 1.8 and 2.0. Sorry, between 1.8 and 2.0 right here. So if I go back here, let me just show you one more time. They want this number to be between 1.8 and 2.0. So we have to divide the absorbance at 260 by the absorbance at 280. So we're going to take a calculator here, and we're going to divide the absorbance at 260, which is 0.51, divided by the absorbance at 280, which is 0.65. and I get 0.78. Yeah. And 0.78 is less than 1.8. So we're going to say no dice. You could have got lucky with this one and just seen that these numbers are smaller than 1.8 and just said no. And you would have gotten lucky and you would have gotten it right. But maybe on the real test, they're actually going to you're going to have to divide those two numbers. Just divide 260 by 280, and that's your number. Okay. Referring to the gel below, what is the approximate size of band B? And they say band B is right here for us. So this band is 1.5 kilobase pairs. That's 1,500 base pairs, because a kilo means 1,000. So 1,000 times 1.5 is 1,500, and here's 1,000. So this one's about 1,400 base pairs, or 1.4 kilo base pairs. So the best answer here, they don't have kilobase pairs, they just have base pairs. So that's our best answer. When using an acrylamide gel for protein electrophoresis. Protein electrophoresis? Yeah, we didn't do it yet. We're supposed to be doing it right now. We're supposed to be running gels with proteins in them instead of gels with DNA in them. So that's called polyacrylamide gel electrophoresis, or PAGE, P-A-G-E for short. And it's the same, it's used for the same purpose as DNA electrophoresis, which is to identify different proteins, because different proteins are different sizes. So if you're looking for a certain protein, you can run a bunch of proteins on a gel and see if you get a band at the size that you expect to see a band at. So anyway, when using acrylamide gel for protein electrophoresis, it's better to use a higher percentage of acrylamide to resolve smaller proteins. That's true. It's just like gel electrophoresis with DNA. The smaller the chunks of DNA, the more dense you want the gel to be in order to get the different sizes to resolve from each other. So like when we do PCR products are super small, so you need super high percentage of gel in order to resolve them. Resolve means separate and see the difference between them. Match the following steps in polyacrylamide gel electrophoresis with the correct purpose of that step. So again, this is just protein gels. And once you understand that it's just protein gels, I think you can probably suss this out. Okay. Staining with some chemical that's blue. That's so you can see it, man. That's to visualize the proteins when you're done, actually. What you do is you stain the whole gel, because the proteins are invisible until you stain them. So anyway, to visualize the proteins after it's done, that's when you stain the gel. Adding loading dye to protein samples. That's just so you can see what you're doing when you're loading it in there. So here, to facilitate loading samples into wells and to monitor the samples moving through the gel. Adding a molecular weight standard to an empty well. That's another way of saying using a ladder or a molecular ruler. And that is to determine the relative size of the samples. And then adding SDS. This is sodium dodecyl sulfate. It's essentially sort of like a super ionized detergent. And it's used when you're doing protein stuff. So it denatures proteins. It messes up proteins. It unravels proteins. So anyway, we want to do that to coat the proteins in negative charge and to denature them. So unlike DNA, which is charged negative, proteins aren't necessarily charged negative. So you have to artificially give them a charge so that the electricity can push them through the gel. And that's what the SDS does, sodium dodecyl sulfate. Yeah. Okay, your supervisor asks you to run a PCR reaction with products ranging in size from 200 to 400 base pairs. That's small, man, compared to like a plasmid, which is still relatively small. Plasmid might be like 7,000 base pairs or 12,000 base pairs. We're talking about 200 to 400 base pairs, small. To confirm the reaction outcome on an agarose gel, which of the following strategies would give you the best resolution? And again, resolution means which one's going to separate them out the best so that you can see the different size bands. You're going to want to use a high percentage of agarose, and you're going to want to use a lower running voltage. So you're going to want to use high percentage of agarose so that you can force these tiny little pieces of DNA to squeeze through tinier holes than normal. And then you're going to want to use a lower running voltage. Otherwise, you're going to melt your gel because there's a lot more resistance in the thicker gel. In agarose gel electrophoresis, the negative electrode is located. So which side do you put the negative, or the black side? You put it closest to the wells. Boom, because DNA is charged negative and negative repels negative. Yeah. Question 13. Assume the DNA in lane 3. They want to see if you know how to read a gel. So here's lane 3 right here. Assume the DNA in lane 3 was loaded after it was digested with EcoR1. If the DNA was originally a plasmid, that means circular, and the restriction digest was complete, how many recognition sites were there on the plasmid for EcoR1? So I'm going to go back to this paper to show you what we're talking about here. If we've got a plasmid like this, and if there's only one recognition site, only one cut's going to get made. And then how many pieces of DNA are you going to end up with? One. That's right. But if there's 2 recognition sites, then 2 cuts are going to get made, and you're going to end up with this chunk here and this chunk over here. So you're going to end up with 1-2 chunks, which is going to end up with 1-2 bands on the gel. So we see 2 bands, which means that there must be 2 recognition sites for EcoR1 up on that plasmid. Okay. 14. A technician is given a sample of DNA from a PCR reaction and a sample of uncut plasmid. The PCR product contains a gene of interest that the technician's supervisor wants cloned into the plasmid. The technician uses software, so that would be like NebCloner, in order to determine which enzymes she can use for each sample. Below is a list of restriction enzymes that cut once in the plasmid's multiple cloning site, and a list of restriction enzymes that cut on both sides. Blah, blah, blah. Here, let me show you something. P-glow, baby. Plasmid. So we use this plasmid a bunch, yeah? P-glow. Boom, here it is right here. So in the P-glow plasmid, an engineer made this plasmid, yeah? Here, let me find one with the multiple cloning site. Let's try this one. Nah. Let's try this one. Okay, so here's a bunch of places restriction enzymes can cut, yeah? And here's green fluorescent protein, and that came from a jellyfish. So someone had to go and cut this gene out of jellyfish genomic DNA, and then they had to paste it into this plasmid right here. So the deal is you need to use the same restriction enzyme to cut it out of the jellyfish genome as you use to open up the plasmid right here. That's the cruche. That's the cruche bit. The reason why is because it leaves sticky ends that are complementary to each other, like ATCG, T, whatever the complement of that is. Whatever the complement of that is. So you need to use the same restriction enzyme to cut it out of the jellyfish genome as you do to open up the plasmid. And the plasmid is generally opened up at this site right here. Plasmids are engineered to contain a cluster of a whole bunch of different restriction sites for a whole bunch of different restriction enzymes, and that gives you flexibility. So you could use any of these restriction enzymes right here in this multiple cloning site in order to cut the DNA, the gene out of the jellyfish genome. So it gives you flexibility. That's what's nice about the multiple cloning site. See right there, it says MCS. That's multiple cloning site. It's just a place that's super dense with restriction sites. So anyway, here's the deal. Here's our choices right here, and actually this is our choices right here, and here's our information right here that we're going to choose. So I'm going to show you two ways to answer this question. First way is the way where you know absolutely nothing about molecular biology. You know nothing about it. What you do when you study these is that the only enzyme, BAM high, there it is up there, but it's not there. EcoR1, there it is there, but it's not down here. Hindi3, there it is there, and there it is there. SAC1, there it is there, and it's not here. So even if you know nothing about this, you can see that Hindi3 sticks out as being different from all the other choices. That's going to be the answer. And if you know the molecular biology, then that's the answer too, because you can use it to cut out from whatever your source of the gene is. In this case, it's a PCR product. And you can also use it to open up the plasmid. Yeah, two different ways to answer the question right. Test taking skills. In recombinant DNA technology, DNA ligase, that's the glue, remember, that glues it together, is used to rejoin which of the following bonds in DNA? You can go into your notebook and bust out that fold out model of DNA and what it looks like, and you'll see that the phosphodiester bond is the bond that links one nucleotide to its 3' neighbor, or its 3' neighbor up here maybe, or its 5' neighbor down here. Anyway, they're linked. The sugar phosphate backbone is linked by a bond called the phosphodiester bond, and that's what the enzyme called DNA ligase does, is it forms that phosphodiester bond. I know this is a lot, you guys, but just remember, you don't have to get an A on this test. You just have to pass it. We're going through it one time. We're going to go through another test another time. You guys are going to be totally sweet. Everything's cool. You guys are going to get certification. It's going to be epic. I'm going to turn this thing off, man.